Thanks, Misty. So you guys are going to have to tell me in the evaluations that Misty and Barbara keep mentioning to you whether this format is useful or whether you would have preferred something different. As we've got the time to expand on targeted and untargeted and workflows, I thought what I would do would be to spend a little time describing what the workflow looks like in the context of an actual metabolomics experiment that I was involved in, or a couple of them that I was involved in. By nature of doing that, it's not going to be necessarily the case that everything that I describe is relevant to what you're going to do in a metabolomics experiment. Everything looks a little different depending on the biological system that you're studying, et cetera, et cetera. So hopefully I'll be able to generalize things where it makes sense to generalize them and thusly help. But sometimes, we're all scientists here, or at least I assume mostly scientists, at least interest in metabolomics if at the very least. And the best way to understand how something is done is to look at an actual example of where it was done, and then talk through the finer points as we go. So let me know if it's not good. If you'd have preferred a different format, then let me know. We'll improve it later. And also let me know, because maybe I can answer your questions or help answer your questions as well as everybody else here, because we don't want you to walk away feeling disappointed. Okay, so where are we right now? We're back on the workflows side of things. Is really by nature a lot of different points along the workflow. Really, the workflow is this whole diagram. I expanded beyond the three at the bottom, which was my talk yesterday: data, sample preparation, instruments, to include some statistical analysis and interpretation and exploration, because when we're looking at actual data, you can't really separate this part from the raw data and have anything meaningful. So I may not take it all the way to the level of, this is biologically a mechanism that I proved, and, I can guarantee you that I won't, but at least hopefully it'll put things in a little bit more context. Okay, so goals for this session. As I mentioned, we're going to work through some real examples of targeted and untargeted metabolomics. You'll probably be sick of hearing the words targeted and untargeted by the time this session is done. Help you understand how to design and experiment around either workflow, or both. That often winds up being the best way to go. And then also we want you to understand the challenges associated with data analysis in both in targeted and particularly in untargeted metabolomics studies. And others will speak to that in even greater detail in more specific terms later on. Okay, so first we'll briefly review the workflows. If the first time you saw it was yesterday, then it helps to take another look at it. We'll look at two case studies. First will be targeted metabolomics in an animal model of metabolic syndrome. This is a research project I've been working on for some time, and we'll show just a small fragment of some of the data today. And the second case study will be an untargeted metabolomics study of bronchoalveolar lavage fluid, which I did with, the project was initiated by Kathleen Stringer, and she'll be here tomorrow to talk to you guys. I don't think she's in the audience here today, about some related work and how it corresponds to some studies she's been doing with NMR metabolomics. But this is some LCMS-based work more exclusively. And in the context of that, I'll spend a little bit of time focusing on this concept of known unknowns. And that's a kind of a funny term that we probably use mainly because it sounds cute, but it describes something that you're going to run into if you do an unbiased metabolomics experiment with us or anywhere else, and you need to be aware of what you're going to get and how to deal with it, what it is and what it isn't. So we'll spend some time looking at that. Okay, targeted metabolomics workflow. You saw it yesterday. We do sample preparation, instrumental analysis. We get our data out of the instrument and analyze it in a straightforward manner. We do calibration and quantitate, and then we do data interpretation and statistics. Sounds fairly straightforward. Let's take a look at an application, and then work through the steps in some detail. I don't want to, don't want this to become a research presentation. That's not my goal. If you're interested in the research, I'm happy to talk more about that, but mainly the methodology and context for you. But that said, I'd like to put the research in at least a little bit of context so you understand why we're doing what we're doing and why metabolomics is a good fit for this research. It turns out that metabolic syndrome, diabetes, obesity are a huge area of research. I would be surprised if a good portion of you weren't working on one of those topics, one of the major areas of research emphasis these days in biomedical field. It turns out that that exercise capacity is a great predictor of overall health, mortality, disease risk, and many other things. This is a classic study in New England Journal of Medicine where they took middle-aged males and measured their exercise capacity and graded it on this term that they called metabolic equivalence. And a higher metabolic equivalent means a greater exercise capacity. And then followed the subjects that they studied for an extended period of time, out to 14 years, and just looked at percentage surviving. They did other things in the study, but just to look at the initial, from the initial cohort, if you had the lower tertile of exercise capacity, less than 5 metabolic equivalents, you had a survival rate of about 65%, whereas those with over 8 metabolic equivalents had better than 90% survival rate. So that's a pretty substantial impact of a single metric, a very good predictor, even better than a lot of other commonly measured parameters, blood pressure, cholesterol, et cetera. I'm not a medical doctor, as you can probably guess, so my terminology is probably a little bit loose in this area, but it's an important metric. And despite decades of study, the genetic and also the metabolic determinants of exercise capacity, and then going along with that, the risk towards diabetes, obesity, et cetera, are not really fully understood. We consider metabolomics to be a really useful and promising tool to help unravel some of these things, or at least move in that direction. Okay, so how are we going to study that? Human beings are a complex system. You can do cross-sectional studies fairly easily. You can recruit subjects, but you don't have much control over the circumstances. You don't have any control over genetics. You just get what you get. That said, the classical way of doing research is to find a particular gene of interest and modify it, knock it out in an animal model, and see what happens. Well, then you get a very specific cause-effect mechanistic type of relationship, but you're miles removed from what really happens in physiology, which is a wide variety of genetic and environmental contributors leading together to a phenotype, and especially in complex diseases like classic type 2 diabetes and metabolic syndrome, if you consider it a disease, are not well reflected by those single very specific mechanistic studies. So we thought to do something that was kind of in the middle, to allow more control than just human cross-sectional studies, but to capture some of the genetic diversity and multi-component origin of these conditions. So some collaborators, some pretty smart people who are now here at the University of Michigan, Steve Britton and Lauren Cope, I set out to some years ago in 1996 to create a model for the genetic heterogeneity of divergent exercise capacity and metabolic function in rats by breeding rats from a heterogeneous stock, this NIH heterogeneous stock, and selecting for running capacity. So a fairly simple protocol. Rats were put on a treadmill and allowed and encouraged to run. And those that ran really well were bred with other rats that ran really well, and those that did not run well were bred with other rats that did not run really well. And their offspring were then tested the same way, and those and successive breeding was done with that with the edges of the population, and it was done in such a manner to minimize inbreeding, so that we weren't trying to create a genetically homogeneous line of rats. The idea was to preserve the heterogeneity, but select for running capacity. And after multiple generations of selection, it's been over 30 since 1998 when this really got up and running full-fledged. We can say that we have two separate lines of rats, low-capacity running rats and high-capacity running rats. And so this is a potentially interesting model to study these genetic determinants of exercise capacity. And metabolic determinants also relevantly. So just to look at why this does this model mean anything? Is it potentially useful? So the low capacity and high capacity running rats differ in many very relevant disease risk factors as they're commonly measured in humans. The LCR are somewhat heavier than the HCR. They have a much higher visceral adiposity. Their random glucose test and their fasting glucose test is slightly elevated. Their insulin is substantially higher, as are their triglycerides. Free fatty acids circulating were higher in the LCR than the HCR, and the blood pressure is a little bit higher. In contrast, the HCRs have evidence, markers of good health. They have higher maximal oxygen consumption by 58% VO2 max, higher whole body oxygen extraction, more capillary density, et cetera, and they live a little bit longer. And this is the result of many studies that have been published on these animals since they were first developed. And so we feel this is a relatively potentially interesting model of human metabolic syndrome. Certainly not perfect. Certainly can't guarantee that all the genetic factors that overlap will perfectly overlap with human propensity towards disease. But it certainly is an interesting place to look. Okay, so here's the real phenotype of these animals. The LCR and HCR rats have more than an 8-fold difference in untrained maximum running distance. So it's important to point out that this is what's done after the rats are just acclimatized to the treadmill. So they know what's coming, but they haven't really been trained. They've been put on, allowed to run, and we just record their maximal running distance. And the LCR rats have from the initial distribution have wandered around in terms of running capacity. They haven't gone down that much. One thing that's important to point out is that animals that refuse to run altogether are not bred with any other animals. So the very bottom of the distribution, just by virtue of the fact that we're scraping kind of along the bottom of how much they can run at all, are left off. So we do have a sort of a floor to the running capacity because, well, many things you can say about that. There's some interest in the non-runners altogether, but running is an important part of the phenotype that Drs. Britton and Koch want to preserve. So these are all running animals, but they remain at fairly low running capacity. And the high capacity runners, on the other hand, have continued to increase over successive generations in running capacity, and by the end of the time, the average running capacity has increased by over 8-fold. So these guys are really impressive when you put them on a treadmill. They are able to run really fast for an extended period of time, over an hour, on a pretty intense protocol where they're running on a 15-degree slope. I don't know if you've ever tried to put a treadmill at 15 degrees and run for a while, but it's hard. And so they do some pretty impressive stuff. Okay, so what makes this an interesting question for metabolomics? Why and why targeted metabolomics? We'll get into that in a minute. Some studies that we did on so you've probably heard of respiratory quotient analysis. This is the sort of thing where you do a treadmill running test and they hook you up to the instrument that measures your oxygen consumption and your carbon dioxide production rate, and you can measure your VO2 max that way. And it also can tell you something about the relative ratio of oxygen consumption and CO2 production. Tells you something can be used to estimate how much carbohydrate you're burning during that period of exercise and how much fat you're burning at any given period of time. And we can do the same thing with the rats. We have a metabolic chamber on a enclosed treadmill connected to an instrument that measures oxygen consumption, CO2 production, and it can measure the relative portion of glucose or carbohydrates being oxidized compared to fat. And it turns out that the low capacity runners oxidize. Initially, the LCR and the HCR aren't all that different. Slight difference. HCR burns slightly more fat, but we can look at that here over in the fat oxidation part. But generally, they're relatively close together. But when exercise starts, the LCR very rapidly switch over to nearly exclusively carbohydrate oxidation, whereas the HCRs maintain fat oxidation and even increase fat oxidation in the early phases of exercise and can hold on to that fat oxidation with reduced carbohydrate oxidation. No, I don't want to say reduced. It's still elevated compared to where it was at rest, but reduced compared to the LCR rats. And they can hold on to that for a longer period of time to maintain their fat oxidation. Eventually, their carbohydrate oxidation goes up. As intensity of exercise goes up, you tend to have to burn carbohydrates because fat oxidation just isn't as rapid at producing ATP equivalents as fat oxidation can be. Okay, so this fuel selection, right? So for some reason, the LCR rats are burning more glucose and less fat. Well, that's a metabolic question. Why is it that these rats can do this? So what we set out to do was to use metabolomics to investigate this more thoroughly. And so here's what our experimental design looks like. This is a picture of our rats on a treadmill. This is the same rat, but really they are different strains, different animals. Low capacity runners and high capacity runners are put on the treadmill for a defined period of rest, exercise, or exercise and recovery. And we have these different periods of time. 10 minutes is close to the exhaustion time of the low capacity runners. 45 minutes is closer to the exhaustion time of the high capacity runners. And after these periods of time, the animals are anesthetized, sacrificed, and we collect their tissues. And we do that as quickly as possible. If you're interested more in tissue collection, I do have a poster on that. I didn't bring it yet, but I'm going to bring it tomorrow. So you can take a look at that. That is an entity unto itself, and I can give you some recommendations about that. I know Anna already talked to you about that yesterday. But anyway, you want to be sure if you're doing tissue collection that you do an appropriate job for what you're trying to do. In this case, we're trying to get as close to the in vivo metabolism. We want to see what happens in response to exercise versus at rest in one group versus the other. So we need really a snapshot of what's going on in the animal. Sometimes when you're doing metabolomics of tissues, if you find yourself in that position, that's not what you want. What you want is to take the tissues out of the animal and put them into a controlled culture condition, and then let everything equilibrate to a sort of a steady state, and then look at differences between one group and another. But here we're trying to be as close to in vivo as possible. And then we extract and analyze the metabolites by LC-MS. And since we have enough time this morning, I thought I'd just show you a little bit of our methodology for the untargeted metabolomics. I'm sorry, for the targeted metabolomics that we're going to go after here. I'm sorry, this slide didn't copy over quite perfectly. For this method, we used a hydrophilic interaction chromatography method. You don't need to worry about these details for any particular purpose. The core lab manages these sort of things. But suffice it to say, this is a method that works very well for polar compounds. We run about 30 minutes per sample, so that gives you some idea of how long a single analytical run will take. Of course, the downstream work on that is a lot more. The setup time and the downstream work is a lot greater. We're running in negative ion mode on our instrument, and we're doing full scanning mass spec, so we're not doing any of the fragmentation I told you about yesterday. This is a targeted analysis, but you can mine the data after the fact for other compounds that fit within the method. Just to show what this method looks like, this method, as I mentioned, is a hydrophilic interaction chromatography, so it retains things that are polar. It tends to work very well for metabolites of central carbon metabolism, intermediates of central carbon metabolism. Cofactors like NAD, aspartate and glutamate are two amino acids that are directly connected to central carbon metabolism. We have a lot of TCA cycle intermediates. We have a lot of glycolytic intermediates that we can quantitate fairly well with the method. We do measure citrate and isocitrate as a combined group, as well as 2-phosphoglycerate and 3-phosphoglycerate. Those are challenging to resolve. We do have some tricks we can use at least to resolve citrate and isocitrate if that's essential, but for these purposes, we measured them as a combined pool, and other metabolites, as you can see there. So this is a good method for measuring central carbon metabolism. There are other methods that are out there that the core lab can use, but this is what we chose for this approach. It can also do a few other classes of metabolites. It can look at free fatty acids and some phospholipids. The coverage is not as good as a lipidomics method would be, which TM will tell you about later today, I believe. But it covers a range of different classes of metabolites. Okay, the sample preparation procedure that we use looks something like this. Again, I apologize, this slide didn't look like this on the previous presentation. I just pulled it over from. But what we do after the tissue is collected is we cryopulverize it, which means we take the solid frozen tissue and grind it up in liquid nitrogen in a mortar and pestle. It's a pretty coarse and seemingly crude and slow process, but it works very well. A nice thing about it is if you're going to be wanting to do multiple experiments on a tissue, if you're going to want to look at proteins or other things, it gives you a nice powder that can be stored in your -80 and then used for any other tests. Whereas if we did a extraction with a solvent, you're pretty much committed to doing metabolomics sorts of studies. You precipitated out all your proteins and you may not recover them effectively for Western blots or whatever else you're interested in doing. After the tissues are pulverized, we transfer them to tubes. We add extraction solvent, and in this case, we're using a mixture of methanol, water, and chloroform. This is, as I briefly mentioned before yesterday, our solvents are mainly organic with some additional solvents as well. It contains a 13C internal standard mix. This is something that's important for targeted metabolomics. If we want to be able to do absolute quantitation, or we want to be able to check the validity of our data for relative quantitation, it's a good idea to have these internal standards present. Our ratio of extraction solvent in this case was 1 milliliter of solvent per 30 milligrams of tissue. So really all you need to do a good job with this assay is 30 milligrams of tissue. And less is workable as well. We can just decrease the volume of solvent. We don't need anywhere near that to analyze. The challenge is it's just hard to come up with reproducible, consistent masses of frozen tissue at very low weights because the stuff is challenging to weigh before it thaws. We have to work quickly in those steps. Anyway, after the solvent is added, we use a probe sonogator to further disrupt the tissue to get everything homogeneous and to lyse the cells and release the metabolites into solution. We centrifuge to pelt the cell debris. The supernatant is directly carried over to LC-MS analysis, and that's the strength of this method, is that we can do analysis without much extra preparation. And then we can dry the pellet, the remaining tissue debris, and use the dry mass for normalization. Alternatively, we could do a protein assay. Both methods are effective. And here we have lots of tissue, so the mass is relatively straightforward. If you have a smaller amount of tissue or cells, then the protein assay works better. Okay, so let's start looking at some data. So first, let's look at some parameters that could be measured using any method, whether metabolomics or not. In fact, glycogen is something that by LC-MS metabolomics is difficult to measure. So this was done by a standard sort of an assay approach. So we have our low capacity and high capacity running rats at 0 minutes, 10 minutes of exercise. After 45 minutes of exercise, there's no LCR bar here because the LCRs can't run that far. They're already exhausted. And then recovery means 30 minutes after exercise, the animals were allowed to rest in their home cage, and then they were sacrificed, and we looked at their tissues at that point, reflecting what happens as the skeletal muscle and other tissues rebuild their metabolites and as they return to a resting state. So glucose is slightly elevated at 10 minutes of exercise compared to rest. At 45 minutes in the HCRs, it's returned to a sort of a baseline level. There's really no differences in terms of blood glucose levels in the LCR and HCR rats. Glycogen turns out to be a really important marker of fatigue, even to a greater extent than other well-known markers of fatigue, although glycogen is well-known in itself. Lactic acid is another sort of classically viewed marker. But glycogen is a carbohydrate reserve existing in skeletal muscle and liver. This is looking at skeletal muscle glycogen in these animals. At 10 minutes, the LCRs have gotten relatively depleted in their skeletal muscle glycogen, so they've used up a fair amount of it, corresponding to the increased oxidation of carbohydrates, whereas the HCR rats are almost right where they started. At 45 minutes, the HCRs have now depleted a good portion of their glycogen reserves, and at recovery, it's on its way back up. Lactate in the muscle, there's some difference. The HCR seem to have slightly higher, although not significant in most cases. The blood lactate is a classical measure of fatigue and of anaerobic, which is a kind of a sloppy term. exercise, but exercise that is not complete oxidation of glucose to CO2 through the TCA cycle. So lactic acid is elevated in the blood of LCR rats at 10 minutes. The HCR rats, it doesn't become elevated until 45 minutes. Okay, so let's look at some data, and I'm just going to scratch the surface on what we've done as far as metabolomics with these animals so as not to create too complex of a story. So we looked at some TCA cycle intermediates, and we were able to measure elevation in certain TCA cycle intermediates in the HCR, though not the LCR rats. So this gives us some sort of a clue that there may be more capacity to support fat oxidation by this upregulation or anaplerosis of the TCA cycle early in exercise in the HCR animals that's missing in the LCR. So here we see it again in malate, and to some extent we do see some elevation in succinate, although succinate is a little bit different. So we have larger error bars. These are experiments in genetically heterogeneous animals. So the data that you get out of it is not as clean as if you had plates of identical cells. That just comes with the territory of working in animals. And you need one thing that's important to point out here is that depending on how reproducible your system is going to be, you need to consider how many replicates you're going to need, even in targeted metabolomics, to get meaningful data. In this case, we had Ns of 6 or 7 rats per group, and we could have done with even larger N in some circumstances. With cells, sometimes if you have very reproducible, an N of 3 is all you need to see large changes. Okay, so the other part of the story that I want to sort of emphasize here with targeted metabolomics is that oftentimes what you'll find will happen is that one piece of the story will lead you to one view on metabolism will lead you to another question and then another study, and you go gathering more metabolites, more information along the way. So TCA cycle metabolites are up, at least some of them are in the HCRs. Well, that may help explain this capacity for anaplerosis may help explain the greater oxidative capacity for fat. But it sort of leads to another question, well, why? So we next chose to look at acylcarnitines, which are a marker of fat oxidation in the blood and in the muscle. Acylcarnitines, it turns out, are basically a part of the process of transporting long-chain fatty acids from the cytosol, which is ultimately coming from the blood, into the tissue, and then from the cytosol into the mitochondria where it's going to be oxidized. So it turns out that long-chain fatty acids are rapidly metabolized to acyl-CoAs. CoAs can't cross the mitochondrial membrane, so they need to be transported by some sort of transport process, and to do that, they're converted to carnitines. Acylcarnitines would be some sort of a long-chain acyl group like C16 palmitate. Complex to carnitine, which is a positively charged sort of head group. And then that can be transported by a transporter across the mitochondrial membrane, where it's reconverted from inside the mitochondrial matrix to a acyl-CoA, where it can undergo beta oxidation and produce energy to generate ATP. So these acylcarnitines, and I think Anna's already mentioned acylcarnitines, and they've showed up in another talk, they turn out to be a good marker of fat oxidation. And you can look at them both inside the tissue, and the skeletal muscle is an indication of what's going on with intracellular metabolism, and also in the plasma. So these things are circulating around in the plasma. They're actually commonly measured in newborns as a measure of, certain ones are indicative of inborn errors of metabolism if you see an elevation in certain levels of particular acylcarnitines. But they're normal endogenous metabolites as well, and they tend to be elevated in the blood when you have incomplete fat oxidation. In other words, the process starts, acyl-CoAs are converted to carnitines, but the mitochondria can't keep up with the fat oxidation, and they leak back out in the other direction through the cell membrane and wind up back in plasma. So this is what we see in the HCR rats, in the LCR rats, we see elevation we actually don't see much elevation. We see slight elevation of the acylcarnitines at 10 minutes. In the HCRs, however, in the plasma, when you go out here to the extended periods of time, at 45 minutes, we see substantial elevation of the plasma acylcarnitines. And so this is reflective, we think, of the extent to which fats have been mobilized and are being utilized in these animals at that long duration exercise. And they're showing up at really high levels in the plasma here because we've begun to reach the point at which they can no longer be handled by the mitochondria. Interesting, however, when you look in the muscle, you see that you see that to some extent in the high capacity runners at 45 minutes. You see something different in the LCRs. At 10 minutes, you don't see much elevation. You do, however, see some interesting changes here in these short-chain acylcarnitines. So what I was describing to you about fat oxidation, that mainly pertains to these long-chain species. But we see some interesting changes in the muscle acylcarnitines. The C2, C3, and C4 tend to be elevated at exhaustion, wherever that occurs, in both the LCR and the HCR. So that at 10 minutes, that's not very dramatic in the HCR. By 10 minutes, it is fairly substantial in the LCR. At 45 minutes here, it is elevated. And so what are these guys doing? Well, they're not really as much byproducts of fat oxidation, although they do occur. They're also associated with branched-chain amino acid metabolism, C3 and C5 acylcarnitine specifically. So that led us to the question of, so we're seeing sort of an exhaustion profiling, some sort of an exhaustion profile associated with these short-chain acylcarnitines. So that led us to the question, well, we know these things are involved in short-chain, in branched-chain amino acid metabolism. They're byproducts of that metabolism. Is there something we can learn about differences between the LCR and the HCR animals by looking at the branched-chain amino acids directly? So that led us to an additional targeted metabolomics assay performed on those species. And this is some of the data that we get out of that. So looking here at valine, leucine, and isoleucine, we do see that at 10 minutes of exercise, we have a statistically significant decrease or comparative decrease between LCR and HCR rats in valine, leucine, and isoleucine. And a lot of that is maybe coming, it's really kind of from both directions. The LCR are slightly, so I apologize here that the direction is a little bit different, but the LCR have elevated level of the branched-chain amino acids, whereas the HCR have decreased levels of these species. And we look at a downstream metabolite through yet another targeted assay, the branched-chain keto acids, which are the first deamidation product of the branched-chain amino acids. And we see that the LCR at 10 minutes of exercise have significantly elevated branched-chain keto acids, whereas the HCR don't get there to the same extent until out at 45 minutes. Interesting that the HCRs at recovery go even higher, whereas the LCR do not. So we're not quite sure what's going on at this recovery time point, actually. But it turns out that branched-chain amino acids may have some substantial roles in supporting fat oxidation capacity. And we have, so we're not the first to describe this. We're the first to notice it in this particular sort of circumstance of exercise capacity. But a recently published paper by Chris Newgard's group showed an association between branched-chain amino acid, insulin resistance, and obesity in humans. So using unbiased, well, this is actually sort of a semi-targeted assay that they did. They used metabolomics to look at a branched-chain amino acid-related PCA factor score for HOMA, which is a measure of insulin resistance or insulin sensitivity. And they found that obese patients have this higher PCA factor score for the branched-chain amino acids. So they're correlated with this insulin sensitivity fairly closely. And so they propose a potential mechanism by which branched-chain amino acids may support lipid oxidation. And here we see showing up again in their pathway, these C3 and C5 acylcarnitines playing a supporting role in branched chain amino acid metabolism. So this is an incomplete picture. It's a complicated model. We don't have a full and complete mechanism for what's going on in these rats, but we found some interesting things. And the workflow that I'd hope to convey through that is that in targeted metabolomics, we get these well-defined pathways that we can look at in some detail. Oftentimes, information from one pathway will lead you to another. And that's so this is just sort of an overall summary looking at the metabolites which are correlated with exhaustion and different between LCR and HCR and exercise. This leads us to further questions which we won't address at this point. Key points for designing targeted metabolomics experiments. I know this was sort of a long excursion into a specific research topic, but to bring it back, the vast majority of metabolites, one way or another, can be assayed by targeted metabolomics, whether LCMS or GCMS or both. But not all classes of metabolites can be assayed simultaneously. So we started with TCA cycle and glycolytic metabolites. We learned that we were interested in acylcarnitines, and that led us to branched chain amino acids and their derivatives. And all of those, in this case, wound up as separate analyses. The good thing that once we're done with that, we have a relatively complete picture of what's going on in all of those pathways. We're not missing much in terms of particular intermediates. So that does increase your time. In some senses, you have to go back, look at your data, understand where you're going, but it leads you in a sequential direction to tell you what's important to look at next. What would have happened had we used unbiased metabolomics for this studies? And we have done that on these rats and other studies a good bit. So we might have found other non-predicted metabolites which would differentiate the LCR and HCR rats. So that would have been a good thing, right? We might have found things that we didn't expect that we should be looking at. Maybe TCA cycle is important, but maybe there's something in some lipid species or some other pathway that we hadn't predicted that would have told us something about the animal. So that would have been a good thing. However, we would have very likely missed some of the metabolites that we did measure in these pathways. We might not have had complete coverage of the carnitines. The unbiased method might not have given us a good measure of acetylcarnitine or C3 carnitine, and we could have missed that important picture that would have led us downstream. It's always possible that you may not have that complete coverage in unbiased. Also, to some extent, the quantitation might be less accurate. That's not as big of a problem as it might seem. In general, untargeted metabolomics is relatively reliable for quantitation, but you can't go back and come up with absolute quantities as easily that can be then referenced back to other animals and other studies. So that's a downside there. Okay, questions about targeted metabolomics before I move on to untargeted, looking at another workflow? Okay, cool. So let's look at untargeted metabolomics, just to refresh your memory as far as the workflow. Same sample preparation and instrumental analysis procedure. We do feature detection first, alignment, recursive processing to validate, to help fill in holes in the data. We're going to do some sort of data reductive analysis, and then feature identification. And then we're going to do some sort of further data analysis, whether it be statistics, pathway mapping, et cetera, to look at this. And so now I'd like to look at this in terms of a unbiased metabolomics experiment that I had the good fortune to be involved with. And some of the participants in this study are present in the audience. Ala, I don't know, you all met her yesterday. She was involved. And just walking into the room, Kathleen Stringer was sort of the initiator of this study. Well, not sort of was the initiator of this study. So I'll direct medical questions if you're interested in the disease phenotype and things like that to her. She'll talk to you later about some other similar work on different studies. So you'll have a chance to hear from her later on. But so the study that I'm going to use is sort of an example of the workflow for an unbiased metabolomics experiment, untargeted, I apologize for using that, the term that we're trying to avoid, is a study that we performed on bronchoalveolar lavage fluid in acute patients afflicted with acute respiratory distress syndrome. So as I mentioned in the previous talk, I'm not a medical doctor. I'm not an expert in these conditions, but my very coarse description of the disease condition is that acute respiratory distress syndrome is a major reduction in lung function, which may be caused by infection or some other traumatic event that's brought on substantially decreased lung function. These are acutely ill patients. They're in the hospital. They have a relatively poor prognosis. Survival rates are, depends on what you're looking at, but they may be 40%, 60%. These are acutely ill patients at substantial risk for mortality. In spite of a substantial amount of study, the actual condition itself is not fully characterized, and there's a lot that's unknown about what causes it, what tells you something about prognosis and treatment. There's a lot of active research going on in that area. So better methods are needed to assess patient condition and characterize the disease. And so it turns out that a common procedure performed on these patients is bronchoalveolar lavage. So this is a picture of a lung X-ray of a patient with this condition. I put it up there. I couldn't tell you from that X-ray that patient has the condition. Someone else presumably could, who is a medical doctor. This is the paper that we'll be referencing on this study. So bronchoalveolar lavage is a procedure which is done to an anesthetized patient who is on a respirator, on a ventilator. It's a procedure which is used to wash out contaminating or the infectious material that tends to and phlegm and other things that tend to phlegm, wrong term. The mucus and other material that tends to build up in the lungs of these patients. Again, I apologize for my very coarse description of the condition. But essentially what's done, and this is just a diagram that I removed from the web, not necessarily exactly the procedure that's followed in our study, but what's done is that a tube is inserted into the respiratory pathways inside the lung, and a saline solution is infused and then withdrawn, and what's pulled out is the bronchoalveolar lavage fluid. And so this is a relatively variable process, although a constant amount of fluid or a normalized to the lung volume can be introduced into the patients. What you put in, unlike plasma, which is tightly controlled and regulated, what you put in and what you get out depend a lot on the condition of the patient, depend on where the tubes are located. So this is something that's more variable than, say, a plasma or a urine sample from a patient. But it's directly assaying the tissue of interest. And that's something that is of definite interest to metabolomics. If you can get closer, plasma is a great biofluid to look at when you're looking at an overall average of what a human or an animal is experiencing throughout their entire body. Because the blood is everywhere, it perfuses all tissues, and the content of the blood is sort of an average over all the organs and systems in the animal. But that's a challenge when you are trying to diagnose what's going on in a particular tissue, because you're dealing with metabolites that are coming from everywhere. And so if you can get closer to the actual tissue that you're trying to study, ideal might be to do a biopsy, right? But those are challenging to do in human patients, and can't be done as sort of a routine diagnostic procedure. So if you have some means of getting closer to the tissue that you're wanting to study, that you expect is involved in a process, then that's a good thing. And so the questions that we sought to answer in this study was, first, is the metabolite content of bronchoalveolar lavage fluid informative of the acute respiratory distress syndrome disease state? Does it have something to say about the disease state? And then a related question, and kind of where we really focus the paper, is can untargeted metabolomics on whatever happens to be in this fluid, not well known, differentiate patients with the condition from controls who are subjected to this procedure but had otherwise healthy lungs? Okay, so this is just a quick look at the demographics of the patient that came out of the paper. We had 18 patients with acute respiratory distress syndrome and 8 healthy controls. This is something to, just because what we're doing here is pointing out caveats and challenges in metabolomics experiments. Sample size is always a concern in these sort of studies, and sometimes you're limited to what you can get. We will do our best in the metabolomics core to provide data that is reproducible as it can be in terms of analytical and technical variations. But when you're dealing with small numbers of patients and the condition is variable, as it was in this case, it can be a challenge to pick out features that meet the rigorous criteria for statistical significance that you have to apply. So it's something to think about if you have the luxury of doing so before the study is even initiated. What sort of study, what sort of sample size are you going to need to get the power that you need to be able to say what you want to say? As Dr. Michaelides was telling you yesterday, sometimes that doesn't wind up working out, and you have a retrospective group of samples that you want to analyze as a pilot experiment, or to just be able to see what you're going to get. We do the best we can, but you have to be aware that there are limitations there. So then you can see other patient demographics. Since this is not a seminar on respiratory distress syndrome, we won't go into the details on the patients and all their lung function conditions. These were patients on a ventilator. The healthy controls were not on a ventilator in the same sense. So there are some differences between the healthy and the ARDS patients that are sort of just a caveat of the study that we're dealing with a very distinct group of patients here, and healthy controls are not under the same level of hospital care as the respiratory distress patients. So these are things that we have to be aware of in interpreting the data of our experiment, and all of these things are characteristic of human studies. The more control you have, the more matched your groups can be, the easier it's going to be downstream to get something out of it. But nevertheless, we move forwards, and this is the schematic from the paper showing the workflow that we used. Basically the same as what I've already shown, so I won't belabor much time on it. We'll talk about this part down here in somewhat more detail as we go. So I won't belabor and describe in detail the LC-MS analysis. We used a multi-platform approach, so we did our classical polar embedded reversed phase unbiased approach as a first pass and with all of the patients. And then we also did sort of a smaller cohort of those patients just to see what we would get using hydrophilic interaction chromatography, the method I already described to you that we'd used for targeted metabolomics. So just because a method can be applied to targeted metabolomics doesn't limit you to using it that way. Oftentimes, if you run it on an appropriate full scan mass spectrometer instrument, you can do either targeted or untargeted metabolomics on effectively the same data. So we use that Hillock method that I already showed you, except we did the data analysis according to an untargeted workflow. And so, okay, all of that's done, right? We'll jump to that step. What do you get out of it? Well, this is a very small portion of what the data looks like when it comes out. And so this is where I should, when you submit samples to the core lab and you do and you want untargeted metabolomics, you're going to get a little bit more than this, right? So to the best of my understanding, at least all the data sets that I've seen previously, we'll do a little bit right off the outset to try to help differentiate groups, one group from another group, to tell you, yes, the method is working, maybe some basic PCA and or PLSDA analysis on your groups if you tell us what your groups were. But the initial analysis that comes with an untargeted experiment is fairly limited because you, as the investigator, really know more about your study, and you're going to know more about what needs to be done downstream. And Ala and others here in this workshop, the goal is that you come away with some idea of, okay, when I get this large table of data, what am I going to do with it? What the heck does it mean? So this though, to say is what it looks like, and so this is much too small. This is about 60 features. There are 1,400 features in this data set from a single ion mode, and we had a couple of different ion modes. Negative ion mode gives a few less features in a typical experiment. But what does this all mean? We see some holes in the data. So let's look at a little bit more closely, just the very first part of the table. We have some sort of assigned sample ID number or metabolite ID, so that are just numbers effectively assigned by the data analysis procedure that we use in the core lab. And what you see on the top of this list is a whole lot of things listed as unknown. So we'll talk more about that in just a second. What we do have for every metabolite is a mass to charge value. That's the ion that was produced in the mass spectrometer. We have a retention time. Here I have, these are the very earliest eluting compounds from the method. These sort of things are sorted just coincidentally in order of retention time. And then we have our samples. So we have control and acute lung injury, also known as acute respiratory distress syndrome, patients. And so these are the patients, and these are the features. We don't want to really call them metabolites yet, but the features that were detected in all of the patients. And so we have some holes, things which, even in spite of recursive processing, we have missing data. And that's another characteristic of untargeted metabolomics. Some features will be present and found in all of your samples. Some features may be missing in specific samples. Maybe they'll be missing in the control samples and present in the experimental group. Maybe they'll be sort of a random distribution throughout your samples. So that's something that you have to deal with on a statistical level because with this sort of untargeted workflow, it's not practically possible to go through manually and look for every single missing peak and say, yes, I can confirm that this is actually missing and it's a zero value and I can put 0 in, or I can put in the lower limit of detection as a proxy for it, or whether it's something that was just in a noisy region of chromatogram and for some reason was missed. So we can do that on a more targeted basis downstream, but we have to be able to deal with it on a statistical level for the massive data set. Okay, so every metabolite has this, very few compounds. So some compounds out of the Untargeted platform, we do have an in-house library that we search all our database against right from the outset. So some metabolites will be ID'd in a data set. Yes, I see a question. So there's a lot of things you can do. Yes, you can do various things. You can say exclude for me when you go to do your data analysis. Exclude anything that has any missing features. That's the most conservative. Probably you'll miss a lot of valid stuff. You can impute a value. You can impute a small value. Not a bad idea would be to use something approximately the lower limit of detection, or some maybe half of the lower limit of detection for that metabolite. And that can be construed based on the other the metabolites that are present in the data set. You can use, there's various means of imputation. K nearest neighbor is one that I'm familiar with that the various programs will use. I'm not an expert in those imputation methods. Suffice it to say that the statisticians that we have in the core lab are, and also programs that all I told you, like Metaboanalyst, which can be somewhat dangerous to non-statisticians, and that you can just check a box and say, okay, I'll try this and see how it goes. They'll let you do those sort of things as well. So there's various approaches that you can use to deal with those missing data. Okay, so this is what the raw data will look like. So what are we going to do with it? Okay, well, first, I apologize. I wanted to spend some time talking about these known unknowns. All right, so known unknown is kind of a strange term. What it means is something that is present consistently in your data set and perhaps in multiple data sets that are seen reliably in all of the features, but we don't have a in-house database match for it yet. So we know it's there. We know we can find it consistently, but it's unknown in the sense that it's unidentified. So what are they? Well, this is the definition of the feature, as I just said. The question is, are they real unidentified metabolites, or are they something else that we don't care as much about? Well, the answer to that question is both. And you're going to see these in your data set. You're going to see a lot of them. Chances are the majority of the features that show up in that massive data table that you may get. And like I said, we're not going to leave you with that massive data table to fend for yourselves. But you're going to want to look at that massive data table because that's where some of the power of untargeted metabolomics comes in. Some of those things that are not identified are in fact artifacts. They're adducts or fragments of other metabolites present in the data, and therefore are not unique chemical entities. That doesn't mean the signal is invalid. It can still tell you about that other entity. It just may mean that when we go to actually do identification on the feature, that it's not something brand new and shouldn't be considered as something separate. On the other hand, some of those features are unique chemical species that just didn't have a match to our in-house library of database and therefore didn't get reported as identified compounds yet. And then the next question that you're going to have is, well, what do I do with it? If it doesn't have a name on it, can I do anything with it? Or do I just let it drop because I don't know what it is and therefore it doesn't have any biological meaning? Well, no. Sometimes these things are worth pursuing. And so my best effort to sort of describe a workflow of what to do with these things was to put together this flow chart. We have a known unknown. Let's just start with any known unknown. By random chance, we pick one. What are we going to do with it? Well, the first question is, the question that we're trying to answer is, do I want to do something with it? Do I want to even bother looking at it and trying to figure out what it is? Well, the first question to ask, is it different between groups? You ran presumably a control and experimental group or something more complicated than that. Do you see some sort of an interesting change in that compound between one group and another? If the answer is no, ignore it. Don't spend any more time looking at it. I know it's nice and satisfying to have a list of, of 1,000 identified features, so you can pat yourself on the back and say, look at all the stuff we measured in the sample, but it doesn't tell you very much if it was identical between your control and your experimental groups. And so that would take a lot of effort. So if it's not different based on the statistical analysis, don't spend any more time on it. Okay, say yes, it is different between groups. My recommendation is that for those compounds you have the core lab, for things that look like potentially interesting, have the core lab look at the raw data. And they can tell you, based on their best opinion, maybe not with 100% accuracy, but they can tell you, well, is this really the unique feature in that signal, in that mass spectrum that we're looking at, and it deserves to be considered a potential putative metabolite on its own right, or does it look like an adduct or a fragment or some other artifact of another metabolite? If it's an adduct, then ignore it. You'll find out what you need to know about that compound when you look at the major species, the identified, presumably, compound from that mass spectrum. Let's say it's not. Okay, well, now it's looking more potentially new and interesting. The next step that we would usually take is search it against an online database, something like Metlin or HMDB, even PubChem if you were feeling like not biasing it. Yes? You can do that, right? And so there is work in the field attempting to to collect the data all together into some sort of a more cohesive strategy to then say all of these things sort of fall under this umbrella, and here's all of the peaks that we have from that. Generally, the quantitation is very comparable across all of the things. You're not gaining much more information by having all of these little fragments. So it's normally sufficient to just mark it as fragment of X, and then leave it at that, rather than trying to sum some sort of signal or average signals between the different compounds. So in general, we'll just sort of leave it there and annotate it as an adduct of X sort of thing, and that's where you would leave it. There is some effort in the field to do this in a more automated process, but it turns out to be challenging. It's hard to put a computer algorithm together that can do the same thing as an experienced mass spectrometrist looking at a spectrum and saying, what does this look like to me? Is this really an adduct? Is it really something that's new, or do we just not know? So annotate, but we don't do anything formal to fuse it together other than marking it as something that looks like an adduct of X. Okay, very good. So we'll search it against an online database if it looks like it's nothing that can be explained as an adduct. Does it have a hit? Does it match some mass in the database? If yes, you may get one compound, and that is great. Okay, so if you get a single compound or maybe a couple of compounds that look like promising putative matches for this compound, then the next step, if you really want to confirm it, is to order the standard or have the core lab order the standard, run it on the same method or a different method, and confirm that it shows up at the same point in the chromatogram, that everything else looks the same as what it did in your sample, and then you have a confirmed identity that you can be confident in. We may already have the standard in our library and it just didn't get matched for some reason, in which case it's even easier. We just look back at runs of that standard compound, and then we can say, yes, this is a good match, and then we know what we've got. If you have a larger list of hits, you have to use some more chemical intelligence to say, what are metabolites that I expect to find in my species? What is this list of 10 hits? What does it have on it? That would be, a common or abundant metabolite. Abundant metabolites are more likely to match what you see in a data set more commonly. But you use your intuition, and you order the most likely candidate. If you don't have it already, you analyze it and you confirm the ID. If it doesn't work out, you take another look at the list of hits and see if it could be anything else, and you keep working on it. So this is a fairly laborious process for some of these known unknowns. A lot of the times for the promising things, it winds up not being that long of a workflow. We can say, oh yes, we already have that. It didn't hit our default database for this reason, but here it is. Sometimes it winds up being an awful lot of work. What happens if it doesn't have a match? If you get a compound that looks interesting, but there's nothing in the library, take some time to think about how important this is. Do you really want to spend the effort pursuing it? If it's really important and promising, it looks like a novel metabolite, it's a highly ranked feature on your list, and you maybe unique among features that's differentiating one group from the other, maybe it really is important and promising to pursue. In which case, you can have the core lab do work to attempt a de novo identification. We'll work to understand mass spec fragmentation patterns. We can think about trying to isolate the thing if it really comes down to it, and using other techniques like NMR. This is a very hard and labor-intensive process. Can be rewarding if you find something really unique and new, but the extent of times, normally this is, for the most part, about as far as we go for most compounds, unless they look really, really interesting. If the answer is no, or there's other things that are very good hits, and I don't want to spend all of my time working on these, real head scratchers, then you ignore it and set it aside. You can report it in a publication as a known unknown of this characteristic. Maybe we can tell you something about its class of metabolites, and we leave it at that level of identification or lack thereof. Okay, here's a quick example in this. This is not the best example. To even call this thing a known unknown, it wasn't a known unknown for long. But we had a feature that was unusual in this acute respiratory distress samples that we don't see in most of our other plasma samples at some level of abundance. And it showed up in some of our samples from the batch, but not all of them. And it was a massive peak. This is the total ion chromatogram. And if something is showing up as this massive in the total ion chromatogram, you've got a lot of something. So we had this peak, and in some samples it just wasn't present. It was it gave a mass charge of 235.18. And the question is, what is it? Why is it in some but not all of our samples? Well, it turns out when you do a METLEN search for this thing, you come up with really one good hit, and the answer is lidocaine. So it turns out that the collection of these samples, and this is something that we actually really would have predicted had we considered how the samples were collected, and was maybe a surprise to the people in the core lab, and me doing the analysis, probably not a surprise at all to the people who collected the samples and initiated the study. Lidocaine is an anesthetic. It's a local anesthetic used to numb tissue, and it's used in the process of doing the bronchoalveolar lavage. For all of the control patients, these are not anesthetized patients. They're not on a respirator, so you have to administer lidocaine. So all of the control patients got lidocaine. The procedure for sample collection, this turned out to be sort of a retrospective study on a larger cohort. So the samples were collected over a larger period of time by multiple technicians. And for the anesthetized, ventilated patients, use of lidocaine is not a critical step. So some of the ARDS patients got lidocaine, and some of them didn't. So it winds up being a big feature in our sample that we can attribute to the anesthetic. Not an incredibly interesting biological feature, obviously, because it's something coming from the procedure. Other things like this happen, artifacts of the sample collection procedure. So that's a nice, easy to identify story. Most known unknowns are not like this. Most known unknowns are much more enigmatic, at least initially. They look like they could be a metabolite. Additional features, feature discrimination and in-depth analysis will tell you whether it's really real and promising, or whether it looks like it's a fragment of something else, or whether it's just chemical noise. And that does happen occasionally too. Okay, so known unknowns. That's sort of my summary there. Okay, so moving back to the data analysis, just to continue on with the workflow, we use partial least, or a la Karnofsky, put together a nice partial least squares discriminant analysis to look to see whether we could find separation in the unbiased data between the control and experimental groups. So this is an example of one of these very simple studies where you have two groups. The discriminant analysis works very well and is clearly able to separate the disease from the experimental group. And the other nice feature about PLSDA, as Alla mentioned yesterday, is it provides a means of ranking your list of features in terms of the features, presumably metabolites, that are most responsible or contribute the most to explaining the differences between the two groups of samples. And these are potential metabolites of interest. So at this level, we haven't done much in terms of identification. We just had a list. Some of these things are putatively identified, some of them not. So let's go back to the workflow. So here's what we've done so far. We took our raw peak list, that big table that I showed you, subjected it to PLSD analysis and feature ranking. Now we have ranked peak lists for all of our different ion modes. The data sets are still separate at this point. We take the top 200, and this was sort of an arbitrary threshold based on what our false discovery rate corrected p-values were giving us. We chose to take about the top 200 features, figuring that this would give us the most interesting features differentiating one group from another from each method, and did a database search. Some of these things were already identified, a fair number were not. And those compounds that had no database match, we selected not to do any further work. We just let them go because they're in that category of features which are either a unknown, difficult to identify adduct or fragment, or there's something that we just don't know what it is, and it's going to take a lot of effort to get there. For those compounds which had a database match, we did more work to confirm the identity for things that we were confident we were able to identify within the context of using standards, not just looking, oh yes, we have hit, saying we go back and run standards or look at standards that we've previously run and things match up. That's the process of validating those features. We then did requantitation. We go back in and manually look at all of our peaks, make sure they're integrated properly, do a good job, and then do another set of statistical analysis on those. Now we're basically sort of in the frame of targeted metabolomics where we have a list of features. And of that, after all of this funneling down, we wind up with 37 confidently identified differentiating metabolites between the groups. So that tells you, part of the reason that we funneled down so extensively is because we have a relatively limited set of patients, and the patients are quite variable. The metabolome in bronchoalveolar lavage fluid is quite variable. So this is really a large degree of funneling from huge number of features to relatively small list of features at the end. It depends on your study. Sometimes you may not funnel down nearly as much through these subsequent steps. And I don't want to say that this is the only means to do a workflow. You kind of have to frame what you do here, whether you take top 200 or whether you take everything and try to do a database search, whether you take only the things that are statistically significant by some threshold after the ranking. All of that kind of has to be determined, can be tailored based on your particular analysis and what gives you the best results. But the basic idea is the same. It's a great idea if you can to go back, if not rerunning your samples in a targeted manner, at least checking the quantitation and validating the features that are of greatest interest to really confirm that you know what you're looking at. And so these 37 identified differentiating metabolites, this is just a table from the paper showing what some of them are. We have a p-value and a false discovery rate corrected confidence value known as a q-value, and we can calculate fold change based on these untargeted data. Some came from the positive mode, some from the negative mode, some from Hillock. Put them all on, and I didn't show Hillock there. So this is these metabolites turned out to be from a range of different pathways. Since we only had 37, they're at fairly disparate points on a metabolic network. So although Alla did a great job taking that data and putting it on pathways, we found that the most logical way to display it for the purpose of our paper was just within a table where we group things based on amino acid, on class of pathways. And this was done through pathway grouping, and Alla can tell you more about that in some sort of detail. But the metabolites that are associated with amino acid metabolism can be broken down into various subcategories, and we see that in acute respiratory distress syndrome, many of these metabolites are up. We see that in terms of lipid metabolism, a good number of lipid species are down, specifically phosphatidylcholines, which turn out to be a sort of a surfactant that's abundant, present in lungs. So we see increase in a good number of these metabolites. Also, we see substantial elevation of nucleotide purine metabolites and pyrimidine metabolites in the acute respiratory distress syndrome. So that there's various different classes of metabolites. What's causing these? Well, it could be a lot of features, right? It could be bacterial metabolism contributing to the content of the lung lavage fluid. It could be cell death and cell lysis, leaking some intracellular metabolites into the fluid. The lack of the surfactants may be indicative of the reduced clearance, reduced ability of the lung to produce these useful surfactants to clear the bacterial contaminants that are present in lung infection and respiratory distress syndrome. So there's a lot of things that can be contributing to this feature. In this case, we were really dealing with just sort of a descriptive work. So we didn't attempt to go further and speculate as to mechanisms as to how you get there. You would need a more controlled system to do a good job with that. But this gives you hopefully some idea of what can be done. And then this is perhaps the smallest pathway network ever from an unbiased metabolomics experiment, but it was just a brief visualization that some of these metabolites are connected. These purine metabolites are directly connected in a single pathway associated with nucleoside breakdown. And this is one of the characteristic hallmarks of ARDS lavage fluid that we detected in our study. Okay, so untargeted metabolomics, to bring it back to the general. Some key points. It offers coverage of a wide range of compounds, both predicted and unknown, but many compounds are not identified, so that's a challenge you're going to have to deal with. More work is required to identify your species of interest. Quantitation may, in some cases, be less accurate. It's not a large problem, but it's something to be aware of. Requantitation of the features of greatest interest can sometimes help you at least do the best job you can. What if we use targeted metabolomics instead for this study? Well, we certainly would have not identified as wide of a range of effective metabolites. We might not have thought to look in purine metabolism as a major differentiating feature, and so we could have missed it. Phosphatidylcholines might have been something we'd chosen to study, but it might not have. So we might have wound up with a very different picture. The data analysis process would have been simpler and faster. If we happened to pick a pathway that was known to be of particular interest, we could have quantitated the metabolites in that pathway and wouldn't have had to go through all the workflow steps, which can take a long period of time, as those who've been involved in these studies can attest. We would have avoided the requantitation and identity confirmation, but the fact that we might have missed the pathways altogether that were really of greatest interest is justification for putting up with these challenges for untargeted metabolomics, at least in the case of this study. Again, just to summarize, hopefully in this, just look at a couple of isolated examples of targeted and untargeted workflows. I've shown you that both are useful and essential methods of assessing the metabolome of biological samples. And where you start, where you wind up, really depends on your experiment. And so we'll say it again, come talk to us. Targeted is straightforward to implement for the most part. There are challenges. Still, it sometimes may require multiple assays. You may find yourself moving from one set to the next to get a more complete picture. Sometimes a single assay is all you need to write the paper, write the study, write the grant that you want to do. Untargeted, on the other hand, the data can be acquired relatively rapidly and in an unbiased manner. But when the experiment is done, the real work is only beginning. The data analysis, the data interpretation, you've got a lot more to do. Known unknowns are a particular challenge, but also a potential opportunity for novel findings. that are associated with untargeted metabolomics. And I guess that's it. I'm happy to answer any questions about workflows or anything else you might have. What's that? So I'm sorry, yes. So we did, at this point, separate between positive ion mode and negative ion mode, as I mentioned. So they're two separate data sets. They ultimately wound up getting integrated together. They're all representative features. Some metabolites will show up in, so as I mentioned, we did a multi-platform approach here. We did, a standard unbiased metabolomics experiment will always contain both positive and negative ion mode studies. So you'll get two data sets. They can essentially be, the reason we keep them separate is for the purpose of database searching. You need to know whether you did a study in positive ion mode or negative ion mode when you go to do your attempts at metabolite identification. So they have to kind of be kept separately. But once you've gotten to the point where you've identified your features, an identified feature is an identified feature. They can all be grouped together. But this was the analysis, the partially squared discriminant analysis was done separately on the positive mode data and the negative mode data. And then we took features from both and identified those that were of interest, took the top 200, identified those that are of interest, et cetera, et cetera. Yeah, all the features thrown right in the analysis. Right. I don't know what the PCA plot would have looked like. I'm sure it would look very well separated because there were big differences. You don't really need PCA at that point to tell you that you have differences, but yeah. Another alternative workflow is to sort of somehow limit the PCA analysis or the PLSDA. You could do PCA at that point if you wanted to, and limit it to the features that look like they may be somewhat determining of some difference between the sample, and in order to get a better separation. Whether that, the power here is that PCA does the work for you of identifying the features that are differentiating and going from there. But there's lots of different workflows that are potentially valid. Talk to people like George and Ala about what is the best way to go for your data set. Usually the first approximation is just to take your raw data set and put it into PCA and PLSDA and see what you get out of it. If you don't get great separation, it doesn't mean there's not meaningful information there. It may just be that you're losing some signal for the noise, and then you need to take those further advanced steps of doing some filtering. Yes. Sure. Yeah. So the question was, the lavage fluid represents a combination of both host and bacterial metabolism, right? And so putting them on a pathway is a little bit of an artificial thing to do because they may be coming from both sources, and we may, to say that they're directly connected is not fair. You're right. You're absolutely right, and that's another reason why we're hesitant to generate an overall metabolic network of this sort of pathway. We operated under for the limited purine metabolism pathway. We operate under the assumption whether they're host or whether they're bacterially derived, and you're right, we don't have the answer for that. They're closely associated with one another. The pathways, the KEGG pathways that are used are reference pathways and are closely linked whether you're in bacteria or whether you're in a mammal. It helps visualize connections between the metabolites, but to absolutely say that this goes to this, and quantitatively so is not a fair means of looking at things. So yeah, that's something you certainly should be aware of, and it may be a little bit of an artificial thing that we did by even attempting to map it onto a pathway. But I think as a means of analyzing data, even in complex scenarios, whether or not we should be cautious to say that by putting things on a pathway, we're not trying to prove a mechanism, right? Or at least that's not always what we're doing. We're not trying to say rate of metabolism is X. We're just trying to visualize connections between changes in the overall metabolome that we're looking To do the more detailed flux analysis and directly prove a mechanism is a much more elaborate process. Sorry, a lot of words to answer your question. Apologize. Anything else?